Hello, everyone, and welcome to today's broadcast, The Role of Vaccination for the Treatment and Prevention of Breast and Ovarian Cancers. It is presented by Keith Knutson, Ph.D., a professor of immunology at the Mayo Clinic. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want, anytime you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Knutson. I will now turn the presentation over to him.
Thank you, Judy, and thank you everybody for your participation in this presentation. Our laboratory has been working on developing vaccine approaches and other immune therapy approaches for the treatment and prevention of cancers for a couple of decades now. Our primary focus is on the development of vaccines for breast and ovarian cancers, which I'm going to spend the vast majority of my time on today. We have some conflicts of interest. Over the course of our discoveries and implementation into the clinic, we license technologies to small companies, enabling them to carry them through, hopefully one day into clinical use in the marketplace. So breast and ovarian cancers represent a worldwide problem, healthcare burden. Across the world, there are a couple of million cases every year of both of the cancers combined, but notably, there's over 650,000 deaths. In the US, we have 240,000 cases of breast and 22,000 cases of ovarian cancer every year, for a combined total of about 54,000 deaths. The costs are tremendous. If you use the Swedish model of estimating costs or from the American Cancer Society, the estimates for breast cancer alone are on the order of about $180 billion. Now that represents about 1% of our gross domestic product and is just a few percentage points less than we spend on our military budget. So you can see that it's a huge healthcare burden to deal with these cancers, and efforts are being made in our laboratory and other laboratories across the US, enabling the development of new approaches for the treatment and prevention. Our focus has been on the identification of strategies to improve the ability of the immune system, which is the body's drug-making machinery, to combat cancer. So one of the unique features of the adaptive immune system is that it consists of specialized cells called T cells or B cells. These are very unique in that each one has unique proteins that help them recognize certain abnormal proteins in the body, such as cancer proteins or proteins derived from microbes, such as bacteria or viruses. In addition, there are different flavors of the immune response, as we've learned over the past couple of decades, in which we can use information that we can use to design effective vaccines for cancer. So for example, there are what we call Th17, Th1, Th2 types of T cells that deal with different kinds of microorganisms and with different types of cancer. And our strategies are aimed at generating specific flavors of the immune response. So we've known now for several years, actually, that the immune system naturally responds to cancer, both breast and ovarian, as well as other cancers. And this is shown here in this particular slide from a colleague that published in New England Journal of Medicine back in 2003 that showed in this instrumental landmark paper that if you have T cells in your tumor, which we designate intratumoral T cells, you have much better outcome in ovarian cancer. And the same goes for breast cancer as well. Our group has shown that this immune response that is associated with either ovarian cancer or breast cancer is antigen specific. So it recognizes specific abnormal proteins on these tumor cells, enabling the immune system to generate immunity with the hope that it can combat it. So the question comes up is, why, if your immune system recognizes your cancer, do the cancers grow out? Well, that's always been a fascinating question for us to try to answer. And through the years, we have identified a number of strategies that cancers use to evade the immune response. And they do so through activating immune suppression. Your immune system is very sophisticated. So in addition to the T cells and B cells, it has a lot of mechanisms in it that prevent it from overacting. In some cases, those mechanisms break down, and as a result, autoimmunity such as rheumatoid arthritis or multiple sclerosis, etc., develop in an individual as a result of the breakdown of these immune suppression or regulatory mechanisms. Well, it turns out that cancer actually uses these mechanisms to hide itself from the immune system. So cancers, despite the fact that they're recognized by the immune system and should be attacked by the immune system, use a wide variety of strategies to prevent. And I'm just showing a couple of examples here. For example, regulatory T cells, or Tregs, which are a specialized subset which suppress the immune response. They get into high tumors in very high numbers. You can see that in that picture on the bottom right-hand side, where you have these regulatory T cells in the tumor. They're blocking the immune system. There's other kinds of cells that also block the immune system. So when we think about how to enable the immune system to combat cancer, we need to think about mechanisms that overcome this immune suppression in the cancer microenvironment. And one way that we can do that is we can develop immune therapies that boost the immune system to very high levels. And that will get many, many T cells and many, many B cells into the tumor microenvironment. Sometimes that doesn't always work, but that is the goal. So we have a number of different approaches that you may come across in the reading of literature or watching shows on TV that tell you about immune-based approaches for the treatment and prevention of cancer. So one of them is cancer vaccines, and these are very much like regular vaccines, and I'll get more into that. But there's also what we call antibodies, and these are produced by B cells. We can make these now in laboratories, and we can give you high doses of antibodies that will combat the cancer. We can also grow your T cells outside of your body and give them back in very high numbers. That's called adoptive T cell therapies. And we also have a number of ways now to reverse that immune suppression in the tumor microenvironment, and that allows for your immune system to really take off and combat the cancer. This has been a very effective approach in recent years and has been approved in a number of different cancers, such as melanoma and lung cancer. One great example is malignant melanoma that was metastatic to the brain in President Jimmy Carter. He was treated with an immune checkpoint blockade strategy that essentially reversed the suppression on his immune system and allowed his immune system to combat the cancer. As a result, the immune system completely wiped out the cancer in his brain, and he's going on to more years of living. So let's get back to cancer vaccines now. There are a number of different choices for how we develop cancer vaccines. We call these neoantigens. So these are new antigens to the immune system. They're proteins. There's a couple of different ways to think about this. For example, there are what we call microbial neoantigens. So for example, if we knew that breast cancer, ovarian cancer was caused by a bacteria or virus, we could just make a vaccine based on that virus or that bacteria. A great example is human papillomavirus vaccine, which prevents cervical cancer. So what that vaccine does is it actually targets the virus that causes cervical cancer. The unfortunate thing is that breast and ovarian cancers don't or are not caused, to the best of our knowledge, by either bacteria or viruses. So we need to think of other approaches. There are, in the green box shown here, a number of other unique proteins that result. I'm not going to go into all of these, but they result from mutations that occur in your chromosomes. And the problem with targeting these specific mutations in the green box here is that they're all very unique to people. And so we can't come up with an off-the-shelf vaccine that we can give to people that would enable them to boost their immune responses against their cancer. So we've come up with a different strategy, and that's the bottom bullet point there, is to target self-antigens and what we call subdominant proteins or subdominant neoantigens. These are normal self-proteins that are in the body, but the tumor cells display them inappropriately or aberrantly. So they're very unique to the tumor microenvironment. And it turns out that your immune system can recognize those. So if we can harness the power of the immune system to recognize these subdominant neoantigens or proteins on tumor cells, that would enable us to generate potentially effective vaccines. Now, for scientists out there, one way to think about this is in this cartoon here. In a normal cell, you have normal proteins shown in blue on the left-hand side being displayed at the cell surface of the normal cell. That's under normal conditions. A tumor cell may express a whole bunch of these different proteins, much higher levels than would occur in normal healthy cells. And as a result of that much higher expression, you get more of these red proteins on the cell surface. Your immune system can recognize that. And if we can identify those, which is what we do in our laboratory, we can actually develop vaccines that stimulate the immune system that can target those red proteins on the top of tumor cells. Here's a great example. We have tumor cells here, and this is a complex experiment, and I'm not going to go into all of it, but we have a complex mixture of cells, T cells and tumor cells, that are shown in the upper two right panels there, where you can see that the T cells, which are showing in the elevated lines on the graph as recognizing and killing, we call that lysis, the tumor cells. But if you take an ordinary heart cell, which is in the two panels just beneath those, you can see now that we can develop strategies that specifically could recognize tumor cells, but not normal healthy cells in the body. We can formulate those into vaccines that enables the body to develop immunity against the cancer. We started this actually several years ago, back in the 1990s. And we used some very old programs to identify what these proteins were. And those proteins are shown in red there. And what they do is they bind the other proteins on the outside of cells. And there's a number of different ways that they do that I'm not going to go into in any significant detail. But we developed a number of vaccines, ECD, ICD, HLA-A2 vaccine, and what we call an E75. Some of these are still used today. I've got a slide that demonstrates that. But what these are doing is they're showing some efficacy in blocking cancer development. One good example is from Brian Zernicki's lab at the University of Pennsylvania, and subsequently he has moved to Moffitt Cancer Center in Florida, where he's shown that if he gives vaccine to patients with very early form of breast cancer, DCIS, then he can get complete shrinkage of those tumors with vaccination, demonstrating the power of the immune response. So we've had numerous vaccines that have been tried, for example, against this molecule HER2-neu that have been developed over the past 10 to 15 years that are in clinical trials. I'm not going to talk about all of these clinical trials. I'm just going to give you a couple of examples that are demonstrating some promise. And so, for example, this is one graph that shows that we can take these vaccines that target these unique proteins, P776, 927, and 1166, and we can generate immune responses by vaccinating individuals in much the same way that you would be vaccinated against the flu virus. And this, in our animal models that develop cancer, shows protection. That's shown in those slides up there where you can give tumors, you can give, mice vaccines, and these mice that develop breast cancer can be protected from developing breast cancer. Now, we have traditionally used vaccines to treat, and they haven't been very effective. Rather, vaccines should be used for cancer in much the same way that you would use a cancer vaccine for preventing disease. And we prevent disease, we use them to prevent disease recurrence in individuals, but there are potentially new ways of incorporating vaccines with a number of other strategies that are being developed, and I'll talk briefly about those. Here's a slide from a colleague of mine at Antigen-Express, which is a company that I consult with from time to time, and they immunized with a vaccine that we developed several years ago, and the patients that are above that red line developed immune responses, and the patients that are below that red line, each shown as an individual symbol, did not develop immune responses. Now look at those red dots in there. Those are the patients that relapse. So you can clearly see in that cohort of people that developed immune responses above that red line, there was very few relapses. And we're seeing this, and this is one great example of the use of vaccines to prevent the redevelopment of cancer after individuals have been treated with normal conventional chemotherapy and surgery. So those were old strategies. They didn't work in everybody. And so over the last 10 years or so, we've been trying to develop vaccine strategies that can be used off the shelf in nearly everybody. Now, I'm not going to go into all of these different permutations and experimental designs that we use, but one of the things that we did is we actually addressed in individuals how well these individual vaccine components worked in the culture dish, and that's kind of shown here. And then we come up with a score that enables us to pick certain parts of proteins that we can formulate into our vaccine. In this particular case, we had 15 different proteins, and we formulated in 4. Those are emboldened in this particular table because they had very high levels of binding to immune recognition molecules in the body. And we subsequently have gone on to employ this vaccine in individuals that have had HER2 positive breast cancer. And you can clearly see on this slide that we can generate very potent immune responses with vaccination in these individuals. But what's important, and unlike the old vaccines that we had developed, we're able to generate immunity in the vast majority of patients. So that wasn't like the old vaccines that I showed on previous slides, where we only saw maybe 40, maybe 30% of individuals developing immune responses. So we have new and improved ways. So this was a phase 1 clinical trial, and it enabled us to determine whether or not the vaccination process was safe in individuals that were apparently cured from their cancer, but at high risk for redevelopment of that cancer at some point in the future. Now, what's interesting is that patients that are on, that have had breast cancer in a special form called HER2 positive breast cancer are given monoclonal antibody trastuzumab, or Herceptin, as it's called in the marketplace. And what we found in those individuals is that they develop an internal or an endogenous immune response against their cancer. So we're looking at ways to improve that. And what's shown here is a graph showing increased immunity on the upper left-hand side, and on the 2 graphs on the right-hand side, we see what we call Kaplan-Meier curves. And those individuals that develop immune responses, which are shown in the red line, have much improved survival compared to those that don't. So that's telling us that we need to boost the immune system in these individuals. And in fact, we just received a grant from the Department of Defense enabling us to test this vaccine in patients with advanced HER2 positive breast cancer in what we call a phase 2 clinical trial. Now, these are very expensive trials, very complex to run. Thus, the grant was $11 million to do this. And we have two arms. We have arm 2 and 3. One of those arms, the ones with the green arrows, that group of patients are going to receive vaccine. We're going to compare those with individuals in arm 2, which is the yellowish orange arrows, and determine whether or not vaccine can improve their immune system or immune response to their cancer, and whether or not it can protect against the redevelopment of the subset of HER2 or breast cancers called HER2 positive breast cancer. So we're working on other strategies too. For example, patients with triple negative breast cancer, a very rare form of breast cancer, are known to develop immune responses against this protein called folate receptor alpha, which is present on the outside of the tumor cells. I'm not going to go into all of the data, but we clearly demonstrate in both ovarian and breast cancer patients that we have high levels of immunity that are generated, and we can try to understand that immunity, try to understand what's being targeted and formulate that into vaccines. We conducted, using that approach in patients with ovarian and breast cancer, we developed vaccines that are as effective in these patients as we saw with HER2 vaccines and HER2 positive breast cancer patients as well. That was just recently published in Clinical Cancer Research. And in fact, we have numerous clinical trials, both in ovarian cancer and breast cancer, in which we're trying this vaccine. And I'll talk a little bit about the results in just a few moments. But needless to say, they're very effective in generating immune responses and have the potential to improve immune responses against the cancer and protect individuals. And I'll talk a little bit about what those trials are on a later slide. But this is just one example of a phase 2 clinical trial in patients after they've been treated with conventional therapy. We're vaccinating them for several months after, and then giving them boosters, and we're seeing efficacy in preventing. Now, there's opportunities also for combining this with antibodies that are used for the treatment of ovarian cancer. So there's one antibody called farletuzumab, which improves progression-free survival and overall survival in patients with a subset of patients with ovarian cancer that are defined by this marker CA125. So those individuals have improved progression-free survival, top right graph, and improved overall survival, bottom right graph. And our hope is that we can combine with these other modalities to further improve immunity in those individuals as well. Now, those are the approaches that we're taking with the non-mutated antigens. But recently, although I told you it's not as easy, we've been starting to target mutations that develop in individuals. And this is a personalized approach. So we're developing in collaboration with a company called Transgene, MyVac. And so each individual that participates in these trials will be given a vaccination that targets their own proteins that are abnormal in their tumor cells. And so this is less off of the shelf, potentially very effective. Now, what this requires is the development of what we call neoantigen discovery bioinformatics pipelines. So we take individuals' tumors and we run through what we call bioinformatic pipelines to identify specific antigens that are unique to that patient. And with all of this power of computers and all of these pipelines, we think that we'll be able to develop a tailored or personalized vaccine for each individual. So those individuals that may not benefit from some of the other vaccine approaches, we can actually now start thinking about what we can look at in their cancers in order to develop a vaccine. So that's going to be something that we're going to be looking at in the future. So I had talked about this idea that immune suppression occurs in the ovarian cancer microenvironment and breast cancer microenvironment, and that blocks anti-tumor immune responses. So we're thinking of developing vaccines that can overpower that immune system. And this is particularly important in ovarian cancer, which has very profound immune suppression in the microenvironment. And if you do manage to boost immune responses with a vaccine, they still have a hard time in the ovarian cancer microenvironment. So we're looking at ways that we can develop vaccines that overpower that immune suppression. So what we've known is that in patients with ovarian cancer, they have high levels of IL-7. Those that have high levels of IL-17 in their tumors have much improved survival. You can see here on the Kaplan-Meier curve on the right-hand or the left-hand side, you can see the light curve showing improved survival in those individuals that have high levels of IL-17. And the reason that they have improved levels or improved survival with high IL-17 is that IL-17 on the right-hand graph is associated with reduced immune suppression. That's called Tregs. And so the higher the level of IL-17 or Th17, as we're calling it in this slide, the reduced immune suppression that they have in their tumor microenvironment. So we've actually developed a novel clinical trial that we're testing right now in patients following primary treatment, in which we've come up with a very specialized vaccine that induces IL-17 and helps the body fight the immune suppression of the tumor microenvironment. Now, this IL-17 inducing vaccine generates IL-17. That's what we want. That overcomes the immune suppression of the tumor microenvironment. So shown on the left hand or the right slide is clearly evidence that we can induce IL-17 in the tumor microenvironment in patients. And using our sophisticated approaches, we're able to develop vaccines that target the vast majority of individuals with ovarian cancer using this vaccine. And so this shows who responds to which antigens contained in the vaccine and how many respond. And as you can see, a high number of patients out of the 19 or patients that we had immunized in this particular trial demonstrate immune responses following vaccination. Now, what's important is that we're out now at about 4 years, and what we're finding with this particular vaccine approach is that 40% roughly of ovarian cancer patients have not relapsed over the course of 4 years. This is an older slide. It shows up to 32 months, but we've gone now nearly 4 years in this trial. So I would have expected, and the literature would have borne out this recurrence rate of approximately 90 to 95%. So we look to be protecting individuals, and we're looking at ways that we can improve recurrence-free survival in these individuals. We still continue to use innovative approaches to identify novel antigens or proteins in the tumor microenvironment. Now, what we know is that the tumor consists of what we call cancer stem cells, and these are what gives rise to new tumors. They're very flexible, so they can either cause a dark blue, purplish blue tumor, which is shown here in this diagram, or they can produce a green tumor. And if you're immunizing against the green tumor, you may not be able to combat the purplish blue tumor. So we're looking at ways that we can identify new antigens that are present on these stem cells. And the reason that that's important, the evidence that we have that that's important, is we see now in patients that depending on the site that is immunogenic in individuals, we see different types of tumors. So the tumors look different depending on where they're at in the body, and that's due to these stem cells. So you can see here in this diagram here that liver and spleen have a certain form of tumor cells, whereas the primary tumor cell are those that are associated with other organs or tissues in the body have a different phenotype or a different appearance to the immune system. So our vaccine strategies may not be complete, and they will need additional antigens in order to prevent recurrence at specific sites. Very complicated, but nonetheless something that we continue to study. This is just a great example on this slide that we have developed vaccines that target not only the tumor cells, but also the cancer stem cells. On the left-hand side there shows that we can generate immune responses to cancer stem cell antigens, but most importantly, we can immunize in just 3 times mice with very aggressive tumors. And as long as we target the cancer stem cell, we can get regression of the tumor. Now, if we stop vaccinating, the tumors will ultimately grow out. We're continuing to look at ways to prevent cancers from growing out in those individuals by targeting their cancer stem cells. So all of the strategies that I talked about, we think are going to be most useful in vaccines that prevent the redevelopment of breast or ovarian cancers. Now, that's the best approach. If you think about the flu virus, if you think about the measles virus, if you think about the polio virus, we don't use those vaccines to treat individuals. We use them to prevent disease. That's where we think. So I think over the next 4 or 5 years, we'll start to get in data about vaccines that can prevent diseases from coming back. And that's going to be particularly useful. And that way, patients can have their immune system stimulated after conventional therapy to prevent the disease from coming back. Because when it comes back, it tends to be very aggressive, or it gets into sites in the body which are really hard to get to, for example, in the skeleton or in the liver or in the brain. And that's how they end up dying as a result of these distant metastases. So I had talked briefly about immune checkpoint blockade when I was referring to President Jimmy Carter and the success that he had, as well as the success of other individuals that have been treated with these kinds of strategies. An immune checkpoint blockade is an approach that uses antibodies, these Y-shaped things that are shown toward the middle on each the right and the left, can block immune regulatory interactions in the tumor microenvironment. So you can see there on the left, there's this Y-shaped antibody called anti-PD-1, and it blocks the interaction between PD-1 and PD-L1. Now, that's not useful in all people. In fact, it's not useful in the vast majority of people, but we do know that some people respond very nicely in some cases with complete shrinkage of the tumors. So in those particular patients, those individuals tend to have very high levels of already existing immune responses. Now, in ovarian cancer, we see very limited activity of these immune checkpoint blockade molecules in ovarian cancer. Much less than we see in malignant melanoma or non-small cell lung cancer. So for example, in this particular study, there was 2 complete responses out of all of the several patients that were treated in this clinical trial. And what we have and others have observed is that the patients that don't respond don't have T cells. So what happens, or what would happen if we combined a immune checkpoint blockade molecule such as anti-PD-1 or anti-PD-L1 with vaccines to boost those immune responses? And this is exactly what we did in this mouse model of breast cancer, in which we immunized mice with a vaccine, and then we gave them anti-PD-1. And I showed you on the previous slide that these animals can actually undergo regression of the tumors, but it grows out. What we observed is that we combined vaccine with immune checkpoint blockade. We have 75% complete regression rate in these animals, and some ultimately redevelop cancer and don't survive as a result. But this is extending the life of these mice from on the order of 25 or 30 days with breast cancer to over 150 days or a half year. So we think that this is an approach that we could potentially use in the clinic to treat patients that have active malignancy. So this is different than the setting of preventing recurrence in individuals, but rather giving them a vaccine and overriding the regulatory mechanisms in the tumor microenvironment with immune checkpoint blockade, leading to improved survival. And this is borne out in their immune responses as well. So this example slide shows that patients that are treated with both immune checkpoint blockade and vaccine develop higher levels of T cells. That's shown in the far right-hand parts of the bars in the graphs there, where you show very high levels of immune responses of flavors, which shown in the pie graph, that are associated with improved survival. So all in all, our strategy has been to develop vaccines for breast and ovarian cancer. We have several approaches that are ongoing. So we have folate receptor, or FR, vaccines to be combined with anti-PD-L1. Those trials are ongoing now. We have standalone vaccines such as FR DC vaccine, triple negative breast cancer FR vaccine, a ovarian cancer FR vaccine, and then we have HER2 new vaccines, which are being used to prevent disease recurrence in individuals. We're also very much interested in the development of vaccines for primary prevention of cancer. And so in the last couple of slides, or in this last slide here, I just want to talk about our efforts to develop a vaccine. So with the National Breast Cancer Coalition, we've come up with a strategy that we've developed over the past few years, which is shown, or the mission is shown in the blue box, which is kind of discombobulated due to some problems with the transmission of the slides. But nonetheless, the strategy or the mission of this interaction with the National Breast Cancer Coalition is to develop a vaccine that targets all of the three major subsets of breast cancer, to develop a vaccine that reduces the incidence of breast cancer, and one that prevents death from breast cancer, and that is safe and cost effective. So in much the same way that you would be given a vaccine for the prevention of cervical cancer by targeting the human papillomavirus, we want to develop a vaccine that prevents development of breast cancer. We have to target these abnormal self-proteins because we don't have viruses that cause breast cancer. And so over the past couple of years, we've refined our understanding of what we need to incorporate in that vaccine, and to specifically develop a vaccine that blocks all three major forms of breast cancer. So we have made this vaccine that targets HER2 new, MAGE-A3, MUC1, survivin, mammoglobin A, and HTERT, which all bind, or which all are expressed in the breast cancer microenvironment, and could be potential targets for vaccines. And this, in collaboration with the National Breast Cancer Coalition, is just getting underway. It's been approved to a certain extent by the FDA for testing in humans. We're still on the pathway to using it as a primary prevention for breast cancer. But hopefully, with the development of this vaccine and potentially other vaccines for breast cancer, we're going to be able to reduce the burden on society that ovarian cancer and breast cancers represent by reducing the incidence of the diseases. And when you reduce the incidence of the diseases, you reduce the need for treatment, you reduce the lost productivity due to death or due to having to go to the doctors or spending time in the hospital, etc. We really want to save lives, and we want to really reduce the amount of resources that are going to protection against breast cancer and ovarian cancer, which I showed way back on the first slide, represents a significant fraction of our gross domestic product. So our approach has been, in general, to develop vaccines that we can give either to prevent cancer or to prevent the redevelopment of cancer. This is how cancer vaccines should be used. This is a change. People have wanted to use vaccines for treatment, but they don't work for treatment. We need more effective clinical settings. So in this particular case, this is showing a graph of a patient that has had ovarian cancer. After we treat them, their tumor volume goes down, of course, with surgery and chemotherapy. We have windows of opportunity that we can continue to boost the immune response, shown in the green line there. And we can do that with vaccines. So more needs to be done in this disease-free period. Let's not wait and see if the cancer comes back. Let's give individuals with vaccine, boost their immune response, maintain that immunity in order to prevent disease from coming back. So we're learning at new ways to develop these vaccines, as I already talked about, particularly identifying the aberrantly expressed proteins and hopefully formulating those into effective approaches that prevent recurrence. We're also learning ways that the immune system is blocking the T cells and B cells in the microenvironment. So we're working on ways of what we call depolarizing immune responses, making them a multitude of different flavors that could potentially work in concert together to eradicate the cancer. We're also thinking about combining with antibody therapy. Antibody therapy also induces immune responses. Can we combine, for example, Herceptin plus vaccine in patients that are at high risk for redevelopment of HER2 breast cancer or ovarian cancer or similar cancers by combining vaccine with monoclonal antibodies? There are also some potential opportunities in the future for employing vaccines for the treatment of individuals with bulky malignancy. But that's not going to be with vaccines alone. We think that's going to be with immune checkpoint blockade. Immune checkpoint blockade doesn't work in the vast majority of individuals because they lack that immune response. We're going to give them that immune response by immunizing them and then treating them with immune checkpoint blockade. Clinical trials are underway to test how do we do that in patients with actively growing disease. One of the things that we need to do, though, is continue to study individuals for immune escape. So some of these approaches, they may seem quite effective, but the tumors grow out. And so some of the strategies that we're looking at as we move forward in cancer vaccine development is trying to understand what new mechanisms the tumors turn on in order to evade the immune response. I didn't talk about that today, but there's a number of strategies that are being developed. So all in all, our approach has been to use vaccines appropriately, either in disease prevention or in combination with immune checkpoint blockade or other types of antibody approaches. We've been very well funded by the Department of Defense for several tens of millions of dollars, the National Institutes of Health, particularly the National Cancer Institute, and we've got a great team at Mayo. I'm not going to go into all of these names, but it really takes a good collaborative team throughout the Mayo Clinic enterprise, as well as individuals outside of Mayo that have come together to support the development of approaches that we think are going to be effective at reducing the risk of death from breast cancer and ovarian cancer. And I thank you very much for your attention today.
Thank you, Dr. Knutson, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, how are cancer vaccines different than ordinary infectious disease vaccines?
Right, that's a really great question. So just to put it very simply, they are different in some ways, but the same in other ways. So vaccines are designed to boost these T cells and these B cells in the body. Those are very unique. So when we use a polio vaccine or an influenza vaccine or a measles vaccine or a tetanus, those are targeting something or proteins that come from the infectious agent. So, and they block viral entry and they cause disease if there's nothing there to stop them. So we're targeting, with infectious disease vaccines, we're targeting microbes, bacteria or viruses. With with cancer vaccines, in contrast, we don't have the same opportunities for targeting microbes. And so we need to go into the cancers and figure out what's different in the cancers. So we can identify proteins which are not derived from microbes that we can stimulate immune responses against. And so while the concept of vaccination to stimulate those T cells, to stimulate those B cells is much the same, the targets that we are choosing for our vaccine are different. We're typically targeting proteins that are present normally in the body, but are deranged in some way by the cancer that the immune system can see. Whereas with infectious disease vaccines, just to reiterate, we're actually targeting proteins that are produced by the bacteria or the infectious agent, but are not normally part of the body.
Thank you for that. Very interesting. The next one is, in what clinical cancer setting are vaccines most likely to be effective when used alone?
Yeah, so as I had mentioned, the use of vaccines has never been to treat disease. We think about influenza every year, and many of us get the flu, and many of us get other types of illnesses that can be prevented with vaccines. But not once, if you have the flu, does the doctor offer you a flu vaccine. And the reason is because by that time, it's too late. And the flu has already started to block the immune system, and in order to run its course. Now, with flu, it runs its course for only a certain period of time. It doesn't want to kill its host. And so it's adaptive ways to, make most of us sick. It does kill certain individuals, particularly the old that don't have immune systems. But the strategy is that you boost the immune system so that a patient has immunity prior to getting the disease. And so in cancer, it's after we've eliminated the disease with surgery, with chemotherapy, then we boost the immune response. And when the tumors start to come back, your immune system remains heightened, just like it does after you get a flu vaccine, such that it can respond very rapidly and overcome all of these immune suppressive mechanisms. And so the clinical setting is to prevent disease, not to treat disease.
Thank you for that. Looks like we have time for one more question. If the immune system naturally sees cancer, why do they continue to grow?
Yeah, so there's a number of different strategies that they continue to grow, despite the fact that the immune system, I briefly alluded to this in the presentation, that they develop all these immune regulatory networks that suppress the immune response. So your immune system is one of the most powerful organs, so to speak, in your body. It can attack specific tissues very specifically and over a very short period of time and completely destroy that tissue. So somebody that has had a transplant, if you don't suppress their immune system, they can completely eliminate that transplanted lung or that transplanted heart or whatever tissue is transplanted. And it can happen against cancer too, but the reason that cancers do grow out is because there are a wide variety of immune suppressive molecules normally found in the immune system. It's very tightly controlled, and different tissues in your body use these mechanisms to prevent the immune system from attacking it. Now, the thing about cancer is it uses all of these mechanisms to block the immune response. And so this is very critical because there's a number of different immune suppressive mechanisms. So when we try to understand a patient's cancer, we try to understand what immune suppressive mechanisms that cancer is using. And even if we do apply something that can be effective at stimulating the immune system and overcoming the cancer, the immune system can the tumor can actually turn on a new immune suppressive mechanism. And so the ability of tumors to go through all of these immune suppressive mechanisms that we have in our body in order to find the right ones to evade the immune system is what happens when cancers grow out.
Thank you. I would like to once again thank Dr. Knutson for his presentation, but also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11th, 2019. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.